I'm going to go ahead and finish off the chapter on the immune system with the last section on HIV and AIDS. So on the left-hand side, what you're looking at is the HIV virus, and this is actually a white blood cell that is being invaded by the HIV virus, which you see in red. Okay. All right, so it's important to be able to distinguish between HIV and AIDS because I find that often there's confusion about it, and we can actually relate it to the relationship between the coronavirus and COVID-19, because it's the same kind of relationship. So AIDS is a disease where the immune system loses its ability to fight off pathogens and cancers. So AIDS is a disease, and the cause of that disease is the human immunodeficiency virus, HIV. So if we want to link this back to what's going on now with coronavirus and COVID-19, the coronavirus, or specifically, is called SARS-CoV-2, because back in 2003, I think you guys weren't even born yet, when I first came to Thailand, there was a breakout of SARS, which is something respiratory syndrome, and I can't remember what it is, but that was actually caused by the virus SARS-CoV-1. So what we're dealing with now is the second iteration of that same virus, and it just happens to have been a lot more infectious than the first SARS-CoV-1. And the disease that is caused by SARS-CoV-2, we call COVID-19. 19 indicating that it started in 2019, not like my esteemed former president, Mr. Trump, said it's because it's number 19. It's just idiot. That's not what it means. 19 stands for 2019 when it was first identified. So want to make sure that we understand the distinction between the virus and the disease caused by the virus. Now, HIV has the ability to divide in several kinds of cells, but its favorite target is the helper T cell. Now, what I want you to understand about the severity of the consequence of an HIV infection, although now I guess there's lots of medicines that are available depending on the country that you live in, the availability of medicines, and how much money you have. People can live quite easily now being HIV positive without actually developing AIDS. And part of what I'll talk about today is how do you know when AIDS starts? But just the severity of the effect of this virus. So if the virus infects helper T cells, put that into context with what you know about the helper T cells and its role in the immune system. So remembering when a macrophage encounters a pathogen, it engulfs the pathogen and displays the antigen on its cell membrane. Then you have the helper T cells, which have to come and try to match with the antigen that is on the surface of the macrophage. If the HIV virus is infecting your helper T cells, then because the helper T cells are what do you call that? What triggers the wrong word I want to use, but I'll just use it anyway because I can't think of it. Helper T cells trigger your entire immune response, meaning your cell-mediated immune response and your humoral immune response. Without your helper T cells, then your immune system is like I don't know what to do, because they're not getting that signal from interleukin-2. Because remember, when the helper T cell matches the antigen for the macrophage releases interleukin-1, which signals to the helper T cell that that's a correct shape. And then the helper T cell releases interleukin-2, which then begins the cell-mediated and the humoral immune response. This is why it can be, and it used to be, such a devastating infection. So HIV enters the cell by attaching to a receptor on the cell membrane of the helper T cell called CD4. It's CD4 and another receptor. This is on the cell membrane of the helper T cell. The infected helper T cell is going to make large numbers of new viruses, because this is how viruses work. You basically have two types of viruses. You have lytic and lysogenic, because I don't specifically have a unit on viruses, because there's just too much to learn in the 3 years. Hopefully, if grade 12 continues to have science in semester 2, which happened last year, then I'll probably be able to fit in more units so we can learn more. So if that's like the new plan, I guess, because last year was the very first year that I've ever taught biology and chemistry for 12B in semester 2. Usually semester 1 finishes and we're done. So hopefully that stays. But anyway, lytic and lysogenic. So lytic viruses are going to infect your cell, take over the cell processes, and then your cell is basically in effect going to produce more viruses that will build up inside the cell until finally it explodes. And when the cell explodes, it releases all of those new viruses that then go on and infect other cells. An example of lytic virus is Ebola. And if you've ever seen any documentaries about Ebola or read anything about Ebola, you'll know that basically what happens is it kind of liquefies the inside of the body to the point where, because the Ebola virus is a lytic virus and it is destroying the tissue, you just start to bleed from everywhere. That's basically what they call crashing, and that's when you're probably going to die, because your nose bleeds, your eyes bleed, your ears bleed, your anus bleeds, everything bleeds, because it's hemorrhaging. If I can spell this right, hemorrhage. I don't know how to spell it. I think it's hemorrhaging. I know there's another H in there somewhere. Anyway, hemorrhaging is when you bleed on the inside. It's not very advantageous for viruses to be lytic, because viruses need a host cell in order to reproduce. So if you kill your host, that's not very helpful. But the HIV virus and most viruses that cause our illnesses that we know of are called lysogenic. And this is when the virus infects the cell and the cell makes new viruses, but the new viruses slowly bud out of the cell through exocytosis. Remember, I taught you that last year, when it goes to the cell membrane and vesicle form, or sorry, it's in the vesicle goes to the cell membrane and fuses with the cell membrane and releases it. So in effect, the host cell stays alive, but it just becomes a virus factory. So just to distinguish between those two, oops, so you can see this. All right, so you can see, I got to turn this back on again. I really wish there was a way for me to erase, but there's no eraser option. So you can see. Okay, so here's the CD4 and the co-receptor. And the co-receptor depends on, I don't know if you can see the text down there. It's called CCR5 if it's a macrophage. But for the helper T cells, which are its favorite target, it's, what is it, CXCR4, whatever. But CD4 is the main one. And then here we go. We've got a helper T cell, which is budding HIV viruses. So these are all new viruses which are being released. And then those can go on and infect a new helper T cell. So how the disease progresses, sorry. If you get infected, the HIV virus, of course, is going to reproduce right after you get infected, because you have a large number of viruses that's going to trigger the immune system to launch a large attack. This occurs within a month of infection. So the symptoms that you would experience would be just like the flu. You would have fever, aches, pains, swollen lymph nodes, and overall fatigue, just being really tired. But I would say 99.999% of people would dismiss this as having the flu. What happens after that, even though the virus continues to replicate, your immune system at this point is able to keep the number of viruses in check. So you essentially feel perfectly normal. So you feel like you had the flu, and then because your immune system keeps the number of viruses in check, you feel fine. So it's just that initial first infection where your immune system's like, what's this? Like attack. And you like launch this massive immune response. That's what causes you to have the flu symptoms. Once it gets the viral number under control, then you feel healthy. So in effect, again, thinking about how you would process what was happening to you, you would just think it's the flu. This second stage can last up to 10 years. Okay, so think about also the fact that if you have unprotected sex with somebody who may or may not know that they're HIV positive, and you are obviously unaware, and then you feel like you have the flu, and then the symptoms go away, and you're like, yeah, I'm totally normal. But meanwhile, you are completely HIV positive, and you continue to have unprotected sex with other people, you are essentially giving those people HIV without even knowing it. That's why it's so important that you have to use a condom for protection at the very least, because even if, like, as females, if you take the birth control pill, that only prevents you from ovulating, which prevents you from being pregnant, but it doesn't protect you from any sexually transmitted disease. The only thing that protects you from sexually transmitted disease is a condom, or I don't think they sell female condoms here, but that would be another option. Eventually, after a decade or so, the HIV viruses are going to start outnumbering helper T cells. So at some point during stage 2, so stage 1 is initial infection. That's when you have a bit of a flu and then you feel fine. Stage 2, during stage 2, again, your immune system is working, is keeping the number of viruses down, and you feel fine. Eventually, towards the end of stage 2, enough of your helper T cells are being infected that your HT count starts to decline. So here is a graph depicting the infection. So you've got primary infection here. On the left-hand side, we have your T lymphocyte count. So this is your helper T cell count. This is the number of weeks. And you see this, there's a really big break here. So stage 1 is here, primary infection. So your HT count goes down, your viral load, which is in red. So you can see HIV RNA copies per milliliter of plasma. The virus spikes, the number of viruses spikes. So acute HIV syndrome, wide dissemination of virus, seeding of lymphoid organs. So you might be like, what the hell does that mean? Okay, acute HIV syndrome, acute just means like for a short time. The opposite of that, referring to disease, would be chronic. So acute means for a short time. And HIV syndrome, that's just that you have symptoms, like that you feel like you've got the flu, high your temperature or whatever, you just feel generally crappy. Wide dissemination means spread. So wide dissemination of virus, it means the virus is replicating quickly and it's spreading everywhere. And seeding of lymphoid organs, that just means that all of your lymph organs are being attacked by the virus, and they're basically becoming centers for viral replication. So this is the part where you feel sick, right? And then you see the viral number drop drastically after that. So this is like the first month and a half, and the number of your HT cells increases. This is called clinical latency. Latent means like a pause, I guess. If I can, yeah, it's kind of a pause, like it's where nothing's happening, like dormancy. So you have this really big break in here because the first 3 months, and then from 3 months and then up to like 10 years here, this is where you have AIDS develop. So this is the part where you don't have any problems, even though over this time your HT count is continuing to drop. But you see how well your immune system is able to keep the viral load in check. That's why these numbers stay pretty low. But after 7, 8, 9, 10 years, that's when your count is so low that the viral load is going to start to increase out of control. And you basically, at this point, you don't really have much of an immune system left. Now, I think it's 200 and that line should be straight. Let's pretend like it is. I think around a T cell count of 200 and below, that is when you officially have AIDS. Now, from that point until death, which I think that in most non-developing countries, people can live with HIV and they don't necessarily die, not like in the 80s where people were just dropping like flies because we didn't know what it was. This virus, by the way, was only discovered in 1988. So that's within my lifetime. It was only discovered in 1988. And then they, of course, they thought it was just like the quote, gay disease, because it was prevalent in the gay population. Not that it wasn't prevalent or not, it wasn't present in the heterosexual population, it was, but through like, you know, fear and just not wanting to, they didn't care about gay people back then, which is horrible, of course, but that's how it was in the 80s and 90s. So, okay, so then stage 3 is when you hit that mark, and that's when officially AIDS occurs. Now you have this thing called opportunistic diseases. I'm going to talk about that in a second. So like I said, the reduction in your helper T cells is disastrous because the helper T cells is what launches your immune response. They have to, your T cells and your B cells, your cytotoxic suppressor T cells, your plasma cells, your antibodies, they don't respond unless interleukin 2 is released. So without helper T cells, you don't have interleukin 2, so you don't have an immune response, okay? Because the helper T cells activate the natural killer T cells, that's NKTC, and your B cells. So as the number of helper T cells continues to fall, your immune system becomes weaker and weaker. So a person becomes more and more vulnerable to other kinds of pathogens that your body would normally be able to combat. And that's called an opportunistic infection, like the opportunity to infect. For example, let's say, I don't know, like a severe flu, or I'm trying to think of some other kind of infection, maybe like a bacterial infection, just anything else that if you were a healthy person with a healthy immune system and your helper T cells were at a normal level, you would be able to fight the illness and you would recover. But for a person that has HIV or AIDS and their HT count is too low, their immune system isn't powerful enough to launch an attack. And that's when you get these opportunistic infections, right? So it is 200 per milliliter. So people that test positive for HIV, they're going to have to go in to get their HT count every, I don't know, I think maybe it depends on what stage you are, what your count is, but I'm guessing on a regular basis, because I'm not a nurse or a doctor or a specialist. I don't know what the exact timeline is, but they would definitely have to go in on a regular basis to get their cell count checked. But at the level of 200 and below, then we say the disease of AIDS officially begins. So this is stage 3. There's 3 stages. So the first stage is the initial infection. You feel a little bit sick, but then you get better. Stage 2 is when your immune system keeps it mostly in check, but over time, the virus is slowly climbing and your helper T cell count is slowly decreasing until you get to this level of 200. And then you have the opportunistic infections and you have a very, very, very weak immune system to try to fight it off. So the question remains, of course, and I'm sure that you've covered it in health class, how is it passed from person to person? It's transferred through body fluids, not including saliva. The most common method of transmission, of course, is sexual intercourse. This includes vaginal, oral, and anal. So using a condom greatly reduces the likelihood of transmission. There is nothing, of course, that is 100% foolproof because condoms are not 100% effective. I think they're around 89 at the most, I think about 89% effective, but 89% is much better than 0%. Again, does not eliminate the risk completely. Another method of transmission is through a syringe. This is the second most common means of transmission. And it's not even just through drug use, but even if, I don't know, you go to some shady clinic that doesn't properly sanitize their syringes, if they use non-disposable, like everybody now uses disposable syringes, but tattoo parlors, that's a really good one. If you get tattoos, you want to go to a reputable tattoo artist. I would just recommend, by the way, never getting a tattoo, because I promise you 1,000,000% you will regret it. Maybe not right away, but speaking from experience, you're going to change your mind and it'll be too late. But because tattoo parlors use, or even piercings the same thing. They use needles and they pierce the skin. And if you do, I mean, you're all your own personal decision. There's nothing wrong with having tattoos. There's nothing wrong with having piercings. I've had both piercings and I have 2 tattoos. I had my belly button pierced, my eyebrow pierced. I've got 4 holes on this ear and 3 holes on that ear. So I'm not a stranger to that kind of thing. It's a method of self-expression. But if you do decide to do these things, then I would strongly advise you, and because I care about you, like beg you to make sure that you source establish like the shop that you're going to, make sure that they have a good reputation. You can even ask them if they have what's called an autoclave. If you're going to like a piercing or something like that, autoclave, it's basically a machine that sterilizes equipment. It basically exposes it to like superheated air and sterilizes it that way. Or if you go to a tattoo parlor, then you make sure that they change the needle every single time. Oh, sorry. Probably a delivery, but anyway. So you just want to make sure that you're careful, because you never really know, I mean, if you're like, because I mean, we're all, well, you guys are still young, but for adults, we were all young and stupid. We've all done stupid things. And don't ever think that if, like your parents or other people, like authorities in your life tell you like, oh, I never made any stupid decisions and I was perfect. No, we're all young and dumb. We all do stupid things. And it's just a fact of life. It's just, that's how you live and learn. But as somebody who wants to educate you, I can at least tell you how you can like be safe and questions you can ask and things like that. The important thing to know, of course, about HIV is that you cannot get it through casual contact. You can't get it from shaking hands. You can't get it from touching the same things that people that have HIV. You cannot get it from toilet seats. You can't get it from sharing bottled water. None of that. Because there was, I don't know, hopefully it wasn't that long ago, but I don't know, maybe when I was university or something like that, there was like people were trying to spread it around that if you sat on the toilet seat when somebody who had HIV, like you could get it that way. No, you can't get it that way. Okay, what about treatments and vaccines? They are trying to create vaccines, and I have not looked at the research for a really long time, but I have, I think I've read somewhere where they're getting really close to finding a vaccine. I do know, speaking of a different STD, the most common HPV, the most common sexually transmitted disease, since I'm talking about HIV, is another virus called a human papillomavirus. I think I might be spelling that wrong. Maybe it's 2 P's. I don't know. Whatever. Virus, it doesn't really matter, but HPV. They do have a vaccine for this one now. And the reason why they have a vaccine for this is because it is the #1 STD of all of the people in the world who have had sex at any point in their life, whether it's once or they're doing it every day, whatever. 80%, I want to say that again, 80% of people that have ever had sex have HPV. It is that common. The typical symptoms for this, if people do develop symptoms, are genital warts. So you get small bumps either on your penis or your vagina. The reason why they wanted to develop a vaccine against HPV is because the majority of males who contract HPV exhibit no symptoms, You would never know that you have it unless you go, like you say to yourself, like, I'm a responsible person and I'm sexually active and I have multiple partners. So I'm going to go to the clinic and I'm going to get tested for STD. So I make sure that my partner knows I'm safe. Most people don't do that. Okay. It's just not, it's not practical. Not that that's not a good idea. It's a great idea, but honestly, it's just not realistic. So for the most part, especially when you're younger, you know, people are going to have sex and you're going to have sex probably unprotected at least once, unless you're like really super good at being responsible, which most young people aren't. No offense, but it's just a fact. Like I said, I mean, most adults aren't responsible either. So who am I to say? Anyway, so the biggest concern is that even among females, I think the percentage is like maybe 50% or something. They are also asymptomatic. So you don't even know that you have it. The problem is that HPV can cause cervical cancer in women. Now, the cervix is the opening to the uterus. So you've got your ovaries, you've got your fallopian tubes here. See my fantastic skills. And then your vagina is here. This is the cervix. Okay. So the virus, because the virus gets deposited during sex in the vagina, it attacks the cells here at the opening of the uterus and it mutates them and it turns them into cancer cells. Now, because there's no symptoms for HPV infection for about half of women, and let's be honest, there's no symptoms for cancer really either, unless like the tumor gets really big and starts causing pain, that women can end up going to the doctor and finding out they have like stage 4 cervical cancer that's already metastasized to like other organs. So again, if you're gonna have sex, be responsible. There's nothing wrong with having sex. It's a normal thing. We're all here because our parents had sex, even though we don't like to think about it. It's a fact of life. Just protect yourself is what I'm trying to say. For females, I would very highly recommend, even though I don't know how it is with new generations now. But when I first got here, girls were like, no, I'm not going to the doctor. No, not unless there's a problem. But you should, especially if you're going to start having sex. I would recommend if you turn 18, even if you're not having sex, go to the gynecologist and have what's called a Pap smear. That's when they scrape. I know it sounds bad. It doesn't hurt. Well, it kind of hurts a little. It's uncomfortable, but it lasts like 2 seconds. They scrape the cells here and they test them just to make sure that they're normal. And you get examined. It's only once a year. So, I mean, you know, once a year, who cares? But it's really, really important because there's so many things that could go wrong and we would never know because there's just no signs of it. Okay. Back to the topic at hand. Vaccines are difficult because the genes that code for HIV's viruses surface proteins mutate often. This is actually relevant to the Delta variant that's going around right now because the Delta variant has 7 mutations in its surface proteins. And I just read an article in the Washington Post. I shared it on Facebook. I'll send it to you online as well. It's easy to read. It's not very complicated language or anything. But the person basically goes through, I think, 3 or 4 past global pandemics that we've had back to, I think, before 1918, but I think 4 different pandemics, what the infection was like when it first came out and what the infection was like about a year later. And they said in every single case, the new variants that come out a year after the original infection are always worse. They're always more infectious because, like I said, a virus survives by infecting a host. So viruses, as they mutate, they become better at being transmissible, at being infectious and contagious. That's why the Delta variant is so contagious. Also, with the Delta variant for coronavirus, at least, it carries a much heavier viral load. So I forget what the number is, but they say that if you get infected with the Delta variant, you're getting a lot more viruses at one time, which then can lead to a more serious infection, which is why we still have to be so diligent and careful and wearing your masks properly, staying at home whenever you can, washing your hands as much as you can. And the biggest problem with developing vaccines against any virus is this mutation. The surface proteins mutate, which means then the shape of your antibodies doesn't work anymore, which means that you have to have a whole new immune response. Okay, that's finished.